Welcome back, friends. Today we are going to study about proteins. Let us start with decoding the word protein. Protein is derived from a Greek word proteus, which means prime importance. So, proteins, the word protein is derived from a Greek word proteus, meaning prime importance. So, the word protein is derived from a Greek word proteus, which means prime importance. Now, protein is an alpha polymer of amino acids. This was about protein derived from a Greek word proteus, meaning prime importance. Let us now see another point about proteins. Protein is a polymer of alpha amino acid. It is a polymer of alpha amino acid. Now, DNL configuration of proteins, or basically amino acids. Let us first give an example of amino acid. An example for amino acid will be alanine, now, a point to be noted about amino acids is that all of the amino acids are optically active except glycine, because glycine is the only one amino acid which do not possess chirality, whereas all other amino acids possess chirality. Now, chirality is presence of 4 different groups around carbon. So we know this is alanine, and alanine, here we can see it has 4 different groups: methyl group, amine group, carboxylic group, and one hydrogen atom. Whereas if we see and compare it with the structure of glycine, so this is our structure of glycine, having carbon, NH2 group, COOH, hydrogen atom, and hydrogen atom. Now here we don't have 4 different groups because this hydrogen 2 are repeated. So this shows that glycine do not possess chirality. That is why it is not optically active, whereas all other amino acids are optically active. Let us see the DNL configuration of amino acids. The DNL configuration of amino acids is determined by the position of presence of amine group. Let us see it with the alanine's help. So this is our structure of alanine, and here if the NH2 group is present on the right side and if it is present on the left side, so this becomes alanine on the right side and this becomes alanine on the left side. So if amine in alanine is present on the right side, it is called D-alanine. And if amine is present on the left side, then it is called L-alanine. D-alanine and L-alanine. So this was the DNL configuration of. So this was our DNL configuration of amino acids. Now let us see if amino acid is kept in aqueous solution, what happens? If amino acid is kept in aqueous solution, Zwitter ion, internal salt structure, or dipolar ion will be formed. Now what do we mean by Zwitter ion? Zwitter ions are those which contain both positive as well as negative, or we can say they are the one containing acid as well as alkali. Like if we have the structure of alanine, and it is kept in aqueous solution, then its structure of Zwitter ion will look like this, having both positive also as well as negative also, acid also as well as amine also. So this is our structure of Zwitter ion, having both positive as well as negative. Now, if this Zwitter ion structure is kept in acidic medium, it forms basic ion, and if it is kept in basic medium, it forms acidic ion. Like if this particular Zwitter ion of alanine, if it is kept in acidic medium, then it will form a basic ion like this. So here forming a basic ion having only positive. And if it is kept in basic solution, if it is kept in basic solution, then an acidic ion will be formed like this. So this way an acidic ion will be formed if the Zwitter ion is kept in basic solution. And on passing the current, the positive one will migrate towards the cathode and the negative one will migrate towards the anode. At a particular pH, it will neither migrate to the cathode, neither to the anode. And that particular pH will be termed as isoelectric point, which ranges from 5.5 to 6.3. That means its pH value is ranging from 5.5 to 6.3. Isoelectric point. So this was about protein. Protein is derived from a Greek word proteose, which means prime importance. It's a polymer of alpha amino acid. And also, when we have studied about the Zwitter ions, when it is kept in any aqueous solution, it forms Zwitter ion, internal salt structure, dipolar ion. And when this Zwitter ion is kept in basic medium, it is forming an acidic ion. When it is kept in an acidic ion, it is forming a basic ion. And at a particular pH, it is neither migrating towards the cathode, neither to the anode, which is known as isoelectric point, which is ranging from 5.5 to 6.3. 5.5 to 6.3. And also we have studied about the DNL configuration that determined by the position of NH2 group. If the NH2 group is present on the right side, it is D. When it is present on the left side, it is N. So this was all about proteins. Next time we will study about the classification of proteins.